Hello, my name is Sangueno. Today I will present about effects of anode current collectors on MFC performance and electrochemistry. The title of the paper is Effects of Wire-type and Mesh-type Anode Current Collectors on Performance and Electrochemistry of Microbial Fuel Cells. This paper was published in the International Journal of Chemosphere in 2018. The presentation is organized as shown on this slide. Introduction. A microbial fuel cells MFCs are an environmental system performing wastewater treatment and renewable electricity production from wastewater. It converts the energy contained in organic wastewater into electricity by using anodic microbial extracellular electron transfer reaction and cathodic oxygen reduction. Innovative wastewater treatment of MFC can relieve the major problems of conventional wastewater treatment processes such as sludge production, odor generation, and energy consumption. To improve MFC performance, a lot of research is going on in various fields. Among them, improving bioanode performance is important, and application of materials with high conductivity, large surface area, high mass transfer rate and high durability is very important. Application of metal current collector is necessary for increasing power generation of MFCs. Carbon felt type anodes can hold a high amount of microbial community and can filter wastewater. And they need current collectors for efficient electron collection generated from extracellular electrogens, exoelectrogen. An optimal ratio of carbon felt to metal current collector and influence of anode current collector on system performance and electrochemistry have not been known yet. In this study, effects of anode current collectors on MFC performance and electrochemistry were tested by using the commonly used metal current collectors, the titanium wire and the stainless steel mesh. Two different types of anodes were made in duplicate with carbon felt sheets by differing in an anode current collector. Performance and electrochemistry were measured in duplicate for each cell. Next slide is materials and methods. First, anode fabrication. Carbon felt for anode material was treated by nitric acid. The procedure is as follows. First, organic residues on the surface of the carbon felt were removed by acetone for 10 minutes, and then the carbon felt was completely dried at 30 degrees Celsius. The dried carbon felt was immersed in 500 milliliters of 5% nitric acid, which was stirred using a magnetic stirring bar for 10 hours at 80 degrees Celsius. The nitric acid-treated felt was rinsed in water to neutral pH and then dried in a 110 degrees Celsius furnace for 12 hours. As a current collector, a titanium wire or a stainless steel mesh was used for each carbon felt anode. Figure A shows a schematic of the carbon felt anode with titanium wire current collectors, and Figure C shows a schematic of the carbon felt anode with a stainless steel mesh. Here, the anode with the titanium wire as the current collector was denoted by CFW, and the anode with stainless steel mesh was denoted by CFM. A current collector was stitched to the carbon felt using cotton yarn as following Figure B and D. The contact areas between a current collector and a carbon felt were 0.6 square centimeters in CFW and 21.7 square centimeters in CFM. Second, MFC construction. A reactor was constructed with a polycarbonate block cut with a cylindrical bed volume. The diffusion layer of the cathode was composed of 60% Fe carbon. An anode electrode was placed vertically very close to a cathode in an MFC. To prevent the short circuit between the anode and the cathode, a non-woven fabric was applied onto the cathodic catalyst side as a separator, and a titanium plate was used as a cathodic current collector. Inoculation and operation. Each anode was inoculated with primary sedimentation tank effluent and activated sludge tank effluent in each MFC. After operating for 6 days in the sewage, each MFC was operated in 50 millimole phosphate buffer solution in a fed batch mode with the 1000 ohm external resistor at 30 degrees Celsius. After 10-day operation, the external resistor was replaced and maintained at 50 ohm. Cell voltage was measured in every 1 minute using the data acquisition system. When the voltage decreased to 20 mV or less, the medium was replaced. One batch took about 24 hours. The electrochemical analysis was carried out from 2-month operation after the inoculation. Electrochemical data presented in the results were collected between the 19th week and 24th week. Next, electrochemical test. All MFC reactor voltages were measured at 1-minute interval using a data acquisition system. Polarization test, electrochemical impedance spectroscopy ICE and anodic cyclic voltammetry CV were performed using a potentiostat. For the electrochemical measurement, the reference electrode was placed very close to the anode in the chamber. All measurements were performed in duplicate. For Coulombic efficiency and energy efficiency measurements, medium samples after 24-hour operation were used for COD measurement. Calculations: power density, current density and optimum external resistance. Coulombic efficiency, energy efficiency was calculated as follows. Polarization resistances of an anode electrode and a cathode electrode and internal resistance of a full cell were calculated from a slope of a linear part of each polarization curve. Results: MFC startup. CFW and CFM were inoculated, and their anode biofilms were enriched at the optimum external resistance in the single-chamber cubic MFC. Their voltages increased to about 60 mV during the first batch. In the third batch, the 50 mm PBS medium with the acetate was used and the MFC voltages increased to 481 MV in CFW and 490 MW in CFM. After 2 batch operations with the PBS medium at the 1000 Ω external resistor, the external resistor was changed to 50 Ω, which is very close to optimum external resistor. During the MFC operation at a 50 Ω external resistor, the MFCS produced 241 MV, CFW, and 232 MV, CFM, and gradually reached 278 MV, CFW, and 288 MV, CFM, in the fourth batch. Thereafter, all the MFCS reached about 300 MV. One batch operation time at the optimum external resistance was about 24 hours. During the optimum resistance operation, CFW exhibited more stable voltage production comparing with CFM. CFM initially produced a relatively higher voltage, but the voltage gradually decreased as the operation continued. Electrochemical tests below were conducted between the 19th week and the 24th week. Polarization test. According to the polarization test, CFM produced more power and current. On average, maximum power density, maximum current density and optimum current density was higher in CFM than in CFW, respectively 32%, 34% and 30%. CFM had lower anodic polarization resistance, full cell internal resistance, and optimum resistance than CFW. On average, anodic polarization resistance, full cell internal resistance, and optimum resistance was lower in CFM than in CFW, respectively 81%, 21% and 21%. However, cathodic polarization resistances were very similar, 51 ohm for CFW and 50 ohm for CFM. The ratio of the anodic polarization resistance to the total internal resistance of CFM was much lower than that of CFW. Among the internal resistance of the MFCS, anodic polarization resistance accounted for 24% for CFW and 6% for CFM, while cathodic polarization resistance accounted for 75% for CFW and 93% for CFM. Impedance of MFC. Anodic ohmic impedance, anodic charge transfer impedance and anodic impedance was lower in CFM than in CFW, respectively 85%, 94% and 92%. A charge transfer impedance was a major part of an anodic impedance for both anodes, accounting for 90% for CFW and 77% for CFM. In the full cell impedance, ohmic impedance and charge transfer impedance was lower in CFM than in CFW, respectively 26%, 68% and 58%. A charge transfer impedance was a major part of a full cell impedance for both cells, accounting for 75% for CFW and 59% for CFM. The ohmic impedance ratios of the anode to the full cell were also lower in CFM than in CFW. The charge transfer impedance ratios of the anode to the full cell were also lower in CFM than in CFW. The ratios of the anodic impedance to the full cell impedance was much lower in CFM than in CFW. The impedance differences between the full cell and the anode were 38.1 ohm for CFW and 20.5 ohm for CFM, in which ohmic components were 10.7 ohm for CFW and 8.7 ohm for CFM, and charge transfer components were 27.6 ohm for CFW and 11.9 ohm for CFM. The anodic CV results demonstrated that CFW generated 41% more current than CFM. The net current productions, I product, were 20.0 mA for CF1 and 14.2 mA for CFM in the anodic CVs. This result is different from the fourth polarization results, where CFM showed higher power and current production. 5. Power and current production during the MFC operation. After the 5-month operation, power and current production in the batch operation of the MFCs at 50 ohm external resistor were investigated. In the voltage profile, the average values of the 18-hour voltage plateaus were calculated. Power and current production during the MFC operation were higher in CFW than in CFM. The average power density and average current density were higher in CFW than in CFM, respectively 51% and 10%. This result is also different from the fourth polarization results, where CFM showed higher power and current production. Next slide is Coulombic efficiency and energy efficiency. C and D were measured and calculated by using the medium samples from the MFCs on day 167. CFW had higher COD removal rate and higher CE than those of CFM. COD removal rate was 11% higher in CFW than in CFM, and CE was 5% higher in CFW than in CFM. However, EE values were very similar, 6.5% for the both. Discussions. First, analysis of the poor performance of CFM in the long-term tests. Although stainless steel has more advantages as an anode current collector material over titanium in terms of both current collection and microbial reaction, CFM showed lower electrochemical performances in the long-term tests than CFW. In the short-term tests, electrical current can be produced by using reactants adsorbed in the carbon felt. However, in the long-term test, mass transfer may be more important because reactant supply and byproduct discharge must be appropriately performed for continuous electrical current production. In CFM, the stainless steel mesh played an important role in the current collection, but mass transfer might be disturbed because it tightly covered the carbon felt. The inside of CFM was covered with the stainless steel mesh, and not only mass transfer but also microbial growth in the carbon felt might be inhibited. However, the small current collector of CFW might not limit mass transfer and microbial growth in the carbon felt. For this reason, CFW showed superior performance over CFM in the long-term tests. Next, engineering implication. If the medium is circulated using a magnetic stirring bar, mass transfer may be enhanced and anodic performance may be improved. Punching holes in the carbon felt can also improve mass transfer and anodic performance. Since the stainless steel mesh covered the inner surface of the carbon felt, it may be a good idea to use a less dense mesh. For the future work, it will be necessary to study the optimal area ratio of the carbon felt and the current collector. 3. Analysis of polarization resistance and impedance. According to the fourth polarization tests, Anodic polarization resistance accounted for 24% for CFW and 6% for CFM. The impedance analysis shows that anodic impedance accounted for 27% for CFW and 5% for CFM. However, cathodic polarization resistance accounted for 75% for CFW and 93% for CFM, and the impedance differences between the full cell and the anode accounted for 73% for CFW and 95% for CFM. This result shows that cathode is a limiting factor in the MFCs used in this study. A charge transfer impedance was a major part of the total anodic impedance for both anodes, accounting for 90% for CFW and 77% for CFM. In the impedance differences between the full cell and the anode, charge transfer impedance was 27.6 ohms for CFW and 11.9 ohms for CFM, accounting for 72% for CF1 and 58% for CFM. This result shows that both anode and cathodes require more effort to reduce charge transfer impedance. However, because cathode is a limiting factor in this study, reducing cathodic charge transfer impedance is more urgent than any other. For this, it is necessary to improve the cathodic catalyst performance. 4. Reliability of this experiment. A few attempts to attach a carbon felt sheet and a current collector. When epoxy glue was applied, the epoxy glue fell off during the anode preparation. When stapler was used, but the carbon felt tore. When stitching was used, the stitched area did not experience any physical damage over time. This was further confirmed by the experimental results demonstrating that the electrochemical performance of the anodes and the MFCs remained relatively constant over time. Relatively large performance differences in duplicate MFCs comparing with those in other abiotic electrochemical systems. In terms of maximum power density, the standard deviation was 77.8% of the average value. Considering that the standard deviations in the previous studies were 510% of the average, we can see the result of normal MFC experiment. Our experimental results are within the normal range. These large performance variations in duplicate MFCs may be due to intrinsic variability of microbial communities. They are composed of many different kinds of microorganisms and various interactions are occurring between them. Even in the same system and the same inoculum, different bioanode performance are commonly observed in an MFC field. Finally, conclusions. In this study, to investigate the influence of anode current collector area on MFC performance and electrochemistry, two types of anodes differing in an anodic current collector were tested in a single-chamber cubic MFC. The carbon felt anode using a stainless steel mesh CFM had a larger current collect than the carbon felt anode using titanium wires CFW as current collectors. In the polarization test, maximum power density, maximum current density and optimum current density were 33%, 34% and 30% higher in CFM than those of CFW, respectively. In the MFC, anodic polarization resistance and full cell internal resistance were 81% and 21% lower in CFM than CFW, respectively. In the ICE tests, total anodic impedance and full cell impedance were 92% and 58% lower in CFM than CFW, respectively. In both electrodes, charge transfer impedance accounted for 90% for CFW and 77% for CFM with the remaining parts of ohmic impedance. However, in the CV tests, CFW generated 41% higher current than CFM. During the optimum resistance operation, average power density and average current density were 51% and 10% higher in CFW than those of CFW, respectively. COD removal rate and CE were 11% and 5% higher in CFW than those of CFM, but the EEs were similar. The stainless steel mesh used as the current collector of CFM might inhibit mass transfer. Although stainless steel has more advantages as an anode current collector material over titanium in terms of both current collection and microbial reaction, CFM showed lower electrochemical performances in the long-term tests than CFW, implying that mass transfer is more important in a long-term operation. In the long-term test, mass transfer may be more important because reactant supply and byproduct discharge must be appropriately performed for continuous electrical current production. In CFM, the stainless steel mesh played an important role in the current collection, but mass transfer might be disturbed because it tightly covered the carbon felt. Thank you for your attention. Don't forget to click that red subscribe button and give me a thumbs.